In this lecture, we will cover other antibiotics that act on the cell wall biosynthesis but are not beta-lactams. So we already seen the role of beta-lactams in preventing cross-linking, and they do that by resembling the diala-diala dipeptide end that gets cross-linked and gets released during cross-linking of the wall. But let's go through once again through the biosynthesis of the cell wall, and then we'll see other possible sites of actions for antibiotics. So the building blocks are constructed in cytoplasm, and one important thing is that, as we know, it contains diala. So in order to have a diala constructed, we need to make the raceme, basically to change the stereochemistry from L-ala to diala, and that we do that with the racemase enzyme. And then these dialas get joined together to form a dipeptide. The dipeptide gets incorporated into a nascent unit that contains NAM and already 3 other amino acids. That then gets translocated into the membrane, in which it gets attached to a lipid carrier and then exposed to the other side of the membrane where NAG gets attached, so the other polysaccharide that is involved in biosynthesis. And then we have the transglycosylation reaction, which attaches two of these monomers together to form the cell wall. And then the cell wall is cross-linked by other enzymes that are the target of beta-lactam amino acids, antibiotics. Okay, so let's start by looking at vancomycin, which inhibits the joining together of these units that contain the lipid carrier, the glyco portion of the cell wall, the peptide portion, including this glycine here, glycine tail. And again, that's what gets attached then to each other and then cross-linked. Okay, so vancomycin looks like this. It's a very complicated structure. We can recognize a peptide chain in the middle. In fact, this antibiotic is made by taking amino acids, modifying them, putting them together, and then glycosylating them. So that's an important part of vancomycin is the glycosyl tail. So how do we make vancomycin, or rather, how do bacteria make vancomycin? So they start by taking amino acids and then modifying them. And so we have chlorination and hydroxylation of tyrosine and phenylalanine. Like for example in this side chain, those then get attached together in the initial heptapeptide. So we have 7 amino acids here. This is a non-ribosomal synthesis, and so happens in the cytoplasm. Once these amino acids have been modified, they get cross-linked in the positions highlighted here in red, forming a very rigid structure. And that rigid structure is very important for the activity of vancomycin. So vancomycin has been discovered in the late 50s and was initially very much used. As many antibiotics that act on the cell wall biosynthesis, it's not particularly effective with gram-negatives. And so we see that we use it nowadays in bacteria that are resistant to beta-lactam antibiotics. We also use it by IV, again for systemic infection, so sepsis caused by streptococcus or other microorganisms that are now resistant to beta-lactamases. The other application we have, because this antibiotic has very poor absorption, so is basically mainly given by IV, is to use it topically in the GI tract. So effectively, the antibiotic is given per pulse, but it's never absorbed nor meant to be absorbed, and it's used for the treatment of Clostridium difficile colitis, which is a very dangerous infection. So let's look a little bit more at how it works. So we have looked at the structure before it was cross-linked. This is what it looks like when the cross-linking has happening. Again, very rigid structure that positions the peptide backbone in this area in a linear position and keeps it, stable basically. And once it's exposed like that, it can form a very strong hydrogen bond to the tripeptide end of the muromyl peptide that makes up the unit, which will be cross-linked in the formation of the cell wall. So this is very important. Again, the mechanism of action is based on that. And more importantly, as we discussed already, what it does is prevents the dimerization of the units, right? So here we have our NAM NAG unit with the pentapeptide, the muramic peptide, and then a glycine tail, and then we'll have another one nearby. This can be either parallel or up and down. Okay, and so what vancomycin does, it basically binds to the end of the peptide and further dimerizes very strongly, and that process prevents then the formation of the cross-linking and the formation of the stable cell wall. So this is very important because the recognition of the specific peptide is super important in the high affinity of the vancomycin dimer. So this is another view of what I just sketched, basically highlighting how the dimer forms. And so it's basically on the other side of the peptide backbone in vancomycin, we have the formation of beta sheet, basically. And at the same time, that allows the interaction with the building block for the cell wall. So what's the mechanism of resistance? Well, the mechanism of resistance is based on the strong affinity of vancomycin for diala and diala. So in resistant bacteria, this diala residue is replaced with a D-lactate, and that causes vancomycin to not recognize it anymore. So what we can have here is the basically weakening of the binding of vancomycin so that the cross-linking reaction can be formed successfully. Okay, so that's an interesting type of mutation that we haven't seen before. Basically, avoiding we have the emergence of resistance by a bacteria that completely changes its mode of action, or at least changes one of the most conserved areas of the cell wall biosynthesis. So one thing we can do also with vancomycin is simplify it, and so we want shorter peptides, still cross-linked to maintain rigidity and to expose the peptide bit into a linear mode, and that's one of the lead compounds that has been developed for recognizing either diala-diala or diala-dilac. Okay, the next antibiotic we'll cover is bezitracin, which is also a very complex polypeptide antibiotic, and is produced by Bacillus species, and specifically by Bacillus subtilis tracy in this case. What it does is that it inhibits the releases of the muropeptide from the lipid carrier, and so the peptide which is attached with muramic acid, basically. And because it decreases the release, then we have that the molecule basically cannot be released outside the membrane. So this is a very toxic antibiotic, which basically Procruz is using systemically. It's also not active orally, and so we use it mainly for topic applications. Other antibiotics that act on the cell wall biosynthesis are the polymyxins, and these polymyxins are also complicated, and they are produced by bacillus species. They're mainly effective against the gram-negative bacterial, and they're also used mainly topically. This class of antibiotics bind to the membrane phospholipids and basically interfere with membrane function, so they're not directly related to the cell wall biosynthesis. I covered them because they're one of the antibiotics used in this very common preparation, Neosporin, and I think many of us have been using it. The idea is to use for topic infections, or to prevent rather topic infections. So we have a combination of bacitracin, neomycin, which is another aminoglycoside, and polymyxin. And the idea is by administering 3 at the same time, we can prevent the emergence of resistance. However, what has been shown that is even with using 3 different antibiotics at the same time, we have the possibility of emerging resistance, and basically excessive use can actually facilitate that process.